Hello everyone. This is the first of the series of the Bioconductor package lectures, and we'll be using the Bioconductor and a mixture of various R commands to analyze the biological data. So for this first tutorial, we'll be looking at the sequin_R package from the Bioconductor, and it contains various functions where you can access lots of biological databases and download it, and you can download sequences, convert it into amino acids, do lots of interesting plots, and analyze amino and gene sequences. So for first installing the Bioconductor package, I have written an function here. Like first, when you install any Bioconductor package, you cannot install using the cran using the cran, because it has a separate repository. The first, you need to source this http bioconductor biosilite and then type the biosilite, the package name. Then it will get installed to you, and then you can again type require or library the package name, and it will get imported. Also, like if you can use that, or I've written this function where it will just type the name of the library you want into this function. It will check whether it is present in the in our package in your computer. If not, it will install it, and again you need to run it, and then it will automatically get imported into your system, right, on the shell. So apart from that, I have also downloaded the ggplot package. You can use here using the tool install packages ggplot2, and then import it into the shell. So first of all, one of the functions of sequin_R is the choosebank package is the choosebank function, where you can see what are the different repositories you can access using the sequin_R package. So let's see choosebank. If I say choosebank info, give me the top four, give me information of the top 4 banks, and they give me information about when was its last updated and what's the release and everything. So it gives you the the right status of updation of different banks you are using, and you can choose one of these banks using like choose bank quotes EMBL. It returns bank name socket and object, and I have already chose bank this one earlier. So so here you can see the condition of this bank name socket. It's showing its EMBL containing like almost 1.8 million sequences, and total species maybe it's 120. I have no idea. So this one, what does that mean? But yeah, so there are also, it shows this number of bases, and 171 sequences, 168 160 subsequences. Okay, so let me choose bank Genbank, because I'll be using the Genbank to query some sequence. You can use the query function here. What does query do? Or type it here in this bar, and to see what the query function does. Actually, it can query using the query function the different databases from the bank you have chosen. For example, I am querying about species Arabidopsis thaliana, and T is the total is the CDS. So it's a complete sequence, and K is partial and not partial. So give me all the CDSs of Arabidopsis thaliana which are not partial, and a not is used as a kind of Boolean operator here. So all the results returns into complete CDS object here, and then you can check the outputs of the values which are returned by the queries. This different this like almost six things. Any LEM returns the number of elements it retrieved from this query. So let's see if I so there are a total of like 158875 number of sequences. So now if I want to fetch the first sequence, I need to use the get sequence function, and from the complete CDS and from the first list. So it will return the first sequence here, and you can see that the sequences are like given as A, T, G, G, C, this something like this, the first 20 sequences. You can also count so the DNA is made up of 4 bases, as you know, A, T, G, and C. So if you use the table command, it will give you it will count the bases for you, and also you can get the name of the sequence using the get name function. So all the get name and the get sequence is in sequenar package. So you can see the name shows something like this. Now I want to plot the dot chart of this of the bases and the amount of and the distribution of the bases. So what I do, I can use the dot chart here, sorry, a dot chart here, and it shows the count of different bases in the first sequence here. So for the codon usage, you can use the UCO function, which will help you to calculate the codon usage. So by codon usage, I mean something like this. So the codon contains these triplets, where each triplet codes for a particular amino acids. So if you're not a biologist, or you don't understand what the triplets mean, so better you study a little bit of molecular biology before doing these functions here. So the triplets like TTT, TTC codes for phenylalanine amino acid. TTA and TTG also codes for the same, or codes for the leucine. CT, all these things codes for the leucine. This 3 codes for the isoleucine. This 4 codes for the valine. So it shows these are the codons, and these are the amino acids which codes for. So once we get that usage, we can plot it something like this. I'm zooming in. So the tryptophan is codes for TGG, and this, it's of no use, it's not used at all here. You can see then this 3 codes for, you can extend it, or you can save it in high definition, high pixels here. So this gives the codon usage, and the maximum is used is, you can see, is leucine, which the maximum usage is. So so get trans will translate the sequence for you, and it will also like, you need to get the sequence, and then you can use the get trans to translate the sequence into amino acids. So here what is happening is collecting the DNA sequence, and it's transcribing it to amino acids, and the table command, I have counted the number of different amino acids is there. So here you can see the histidine, there are 3 histidines, and methionine is 5, isoleucine is 5, serine is 4, glycine is 4. So all these things gives you the list of total amino acids, and that can be coded from the gene sequence. So AAA converts the single letter amino acids into the 3-letter amino acids, and then you can just spot here. This is the stop codon, and all the amino acid counts. This, like, glutamate is the maximum here, and leucine is here maximum. So now I'll be showing you to use the SwissProt resource. Then you can choose the SwissProt database using the ChooseBank SwissProt. So I have chosen the Swiss prot here. So you can query using this. This EC is the Swiss prot accession number is, and you can get anything. You can search anything using Swiss prot accession number. Just go to Swiss prot and try to find out which accession number you want to use, or if you have a list of accession numbers, you can give it here, and you can get that sequence. You can get the sequence from the Swiss prot. So I have queried then the lepra, and I'm getting the sequence here. So I'm querying ulcerans. I'm getting the sequence here. I'm closing the bank and dot plotting using the dot plot. So dot plot is one of the methods of comparing two sequences. I remember it was the first of the methods of comparing two different kinds of sequences. Is used kind of mostly in the similarity based methods. Apart from that, you can also use a local FASTA file. I have collected a bunch of sequences in the test.fasta. So I have put my test.fasta in my this folder, in the data folder here. So I loaded it, and remember the sequence, since you are importing amino acid sequence, so the sequence type you need to put is AA, which recognizes it is amino acid. If you don't put it AA, it will recognize as a nucleotide sequence. So length is shows it's 4 length, and shows the different types of list which is there. Means there are 4 sequences. So this one gives you the second one, the second sequence. The name is this PDBIDJN sequence, and is attribute of class sequence FASTA. Now if I want to convert the names to amino acid 3-letter words, I have converted this, and you can so you can use the table and tab order, then it will show you the amino acid counts here, something like this. You can also compute the isoelectric point of the protein sequence and the molecular weight using compute PI and PMW. You can also create hydropathy plots, like how much hydrophilic and hydrophobic your proteins are. So for that you have the sequin. Go to the vignette of the sequinar, where you can get the hydropathy values, and actually it comes with the package, and you can import the hydropathy values of each amino acid. You can get using in the EXP data set. See, I have found like everything is now is given here, and it's been ordered from higher to lower to higher, and then you can plot. So once this is ordered, you can run this function, and then just give your sequence, which is A1, and the coefficients in the KDC, which is this one, and you can get. So first, what I did, I collected all the names and I ordered it, and then I wrote a function of where I give the data and then the coefficient values, that is the KDC. So actually it maps your amino acid sequence to the coefficient values and get all the values, and creates a data creates a data frame. So it returns a data frame something like, which is A, like it returns all the values of the sequence, and then you just convert your amino acids, this thing, so this corresponds to this letters, and then I just plot it here. It shows how much hydropathic your uh, proteins are. The one is kind of a standard measure. If your protein shows this region is something like, so this region, mostly if you see, this region is hydrophobic region, this one is hydrophilic, this one is strongly hydrophobic regions. So there you can get the most sequence elements which shows hydrophobicity. uh, Now coming to if I want to search patterns from the Genbank, so what I need to do, I need to install this biostring package, and then I already chose Genbank. I made a query. It will take some time to query the Genbank database. So I'm querying Homo sapiens and protein kinase, and @ is the wildcard character. So give me all the gene sequences containing Homo sapiens, and where the wildcard is PRKA, which the gene name is PRKA, @ is the wildcard character. So PKA, you can see here it gave me the all. So C2S is so for you have seen here, like the PKHS returns the single letter characters. So C2S converts the single letter characters to full string here, the C2S command. And then I collect, this is the first sequence, and suppose for example this is the pattern I want to search for in the PKA. I need to use the match pattern function from the biostrings package. So here it goes. When you do it, it shows which are the positions where your this string can be found here. Okay, this completes the first of the series of lectures of the bioconductor. Next we'll be showing you how to code the sequence alignment programs, like the dynamic programming algorithms, and compute sequence scores and align sequences. I hope you enjoyed this video. Thank you.